Good morning, everyone. Welcome
Good
you
morning.
for the third session of the Wildlife Disease Investigation webinar series that we are doing. And I am Daniel from Life Trust India. We are into wildlife conservation promoting activities involving community in India and other regions also. As I mentioned, this is the third session, and all the sessions will be recorded. The recordings of the previous sessions will be available through YouTube. We'll be sending the links of the previous presentation and also the current presentation maybe in a couple of days. Okay. If you have any questions for the presenter, you are most welcome to post your questions on the text box, and then all the questions will be answered at the end of the presentation. Okay, with this, the certificate of participation will be issued to all the participants at the end of the series. So we have 3 more sessions to go, and so that's the thing. So now I request Heidi to just introduce the presenters, and over to Heidi now.
Thank you, Daniel. First of all, I would like to welcome everyone on behalf of Asian Elephant Support, who is one of the partners in this series of webinars. And we hope that you not only enjoy the webinars, but learn something from them. And we also encourage you to ask questions and to keep in touch with our presenters over this year and the number of webinars that we will be doing on various topics having to do with wildlife disease investigation. So this webinar is primarily about elephant health. Our first presenter is Dr. Christopher Stremme. Dr. Stremme is a veterinarian and a former elephant handler and trainer with over 25 years of experience in elephant healthcare and management. For the last 20 years, Dr. Stremme has been involved in various captive and wild elephant welfare and conservation projects in several Asian elephant range countries. Since 2004, he is living in Sumatra, Indonesia, where he has developed and managed elephant healthcare programs with local NGOs, providing regular healthcare and management assistance for captive and wild elephants throughout Sumatra. In 2015, he joined the veterinary faculty at the Syakuala University in Banda Aceh as an associate lecturer to lead the development of elephant and other wildlife veterinary training and teaching programs, as well as the development of a wildlife ambulance. Since 2016, Dr. Stremme is the program manager for the organization Community for Sumatran Nature Conservation, working on elephant conservation programs in the Waikambas National Park. Today, Dr. Stremme's presentation is about some healthcare aspects in Sumatran elephants Elephas maximus sumatranus. So Dr. Stumme, over to you.
Thank you very much. Thank you for the introduction, Heidi. And I think with no further ado, I try to share my screen. Let's see if that is working. And so is my screen shared with the first slide?
Nope.
Huh? No?
Not yet.
Shit,
Not
I
yet.
don't know what I did wrong. I clicked on share screen. I click on share screen, then still not shared.
Yes, now there we go.
There you go. Okay.
Now we can see it. So you can, if you start the presentation at the first slide, then, yep, there we go.
Now we're full screen. Okay. Thank you very
Yes.
much. Welcome everybody. I'm working with the veterinary faculty at the Sierra Kuala University, where we have wildlife ambulance, through which we are involved in the regular veterinary care for a captive elephant population of about 100 elephants in different locations. And the major part of the healthcare is actually preventative healthcare. Only maybe 10% of our activities with the captive elephants actually curative care. Mostly it is preventative schemes, which I want to mention the most important. So regular health checks, training the elephants for medical procedures, foot care, anti-parasitic scheme, and tetanus vaccinations. For the regular healthcare, it is important that we are not only come to the elephants if they are actually sick, but we regularly every 3 months visit each of the places and do general health checks in each of the elephants, like doing a body condition score, weighing the elephants, measuring the elephants, once yearly taking also a blood sample and doing a basic clinical examination that gives us an overview and helps us to be ahead of any problems or anticipate any problems that may occur. To be able to do that, for us, it's very important that the elephants properly cooperate, and they only do that if the elephants are trained for that. So when I started to work in Sumatra, most of the elephants were not really specifically trained for veterinary procedures, and sometimes it was very difficult to approach the elephants for any kind of treatments or examinations. So part of our regular work with the mahouts is to encourage the mahouts, to guide the mahouts, to train the elephants, specifically also for different kind of veterinary needs of care needs. Like one is, for example, having the elephants trained to being in a restrained shoot. So the elephants can be restrained. They're used to that. They're not stressed when they're restrained. Either they're on chains or they're in a restrained shoot. The elephants know that and easily accept that. That makes it a lot easier, alongside with training the elephants to be adapted to get touched and examined at all parts of their bodies, whether it's the ventral abdomen, whether it's between the leg, around the eyes, whether it's on the feet, or also being able to manipulate the trunk, touch the trunk, doing examinations, any parts of the body. Also training the elephants for accepting and knowing that samples are taken, like blood samples are taken without being the elephant stressed. And for example, also doing oral examinations of the mouth cavity. And especially for captive-born calves and for rescued orphans, we very much encourage and guide the process to start the training very, very early with the basic training needs. At an age of 3 to 4 months, the basic training is started with these young elephants, which is a lot easier and a lot less stress and adapts these procedures at a very early age. One part, as I said, is regular foot care. So why is foot care sometimes needed in captive elephants and not in the wild? Well, in the wild, elephants are active for 15 up to 18 hours, sometimes even more, walking around looking for food, migrating in the habitat. In captivity, elephants are active usually a lot less, or mostly a lot less, which can lead to the fact that foot pad and toenail, which are permanently growing, overgrow, and the anatomy and the function of an elephant's foot is quite different to a horse. So the weight is distributed over the entire foot pad through this digital cushion made from collagen fiber and connective tissue. And in the opposite to a horse, which walks on the tiptoe, the weight of an elephant is distributed over the large footpad, and only very little, if none, weight is going on the toenail, which is actually a much smaller and a less stiff and rigid structure compared to horses. If due to lack of exercise, this is starting to overgrow a lot, when the elephant is walking, a lot more weight is bearing on the toenail, abnormal weight, and that can lead to cracks in the toenail. You can see here a very intensively overgrown and cracked toenail, and these toenails can split through the nails and can actually cause splitting into the sensitive issue, causing literally open wounds, can also get infected. Overgrowing toenails also can enclose the space between the toenails. Usually there should be an open space between the toenails, so the toenails, when the elephant's walking on the foot, is flexible. If that is not the case, so the toenails meet in the middle and they can break, and also can cause create cavities and holes which entrap dirt and foreign bodies, which can lead to destruction of the lower tissue or deeper tissue. The same can happen with a footpad. Footpads also can develop excessively if the elephants are not walking enough, and developing double layers, which in between double layers also some dirt, some foreign bodies can be trapped. Just try to look here, like here, and that can lead to destruction of the footbed. This is why we when we're visiting, we're doing foot care together with the mahouts. We also encourage them to do the mahouts by themselves on a regular basis to prevent these problems, because once you have something starting, you'll see here 2 pictures from the same toenail, 2 years apart from each other. Yeah. And nothing has been done, no interference has been done, no trimming has been done, and the crack has cracked through up to the top and has caused a wound on the foot, an infected wound on the foot. Also, these bad cracked nails can be treated, but it takes a long time. Here's an example: completely split through nail, cut apart in 2 halves. After the initial trimming, that is what it was looking like. Yeah, you can see the crack went deep inside, into the vascularized area, and this is not done with one intervention. So in a several months or weeks intervals regularly, you have to retrim that to ensure the toenail is kept short and no weight is bearing on the toenail, and then the toenail starts regrowing from up from the top to the bottom. And after a period of more than a year with regular retrimming, the toenail is healed, and this is the condition. Can you see my cursor here? Is that visible on the screen?
Yes, Christopher, we can.
Yeah. So you can see that is after the last trimming, after over a year. So this is completely healed, but it takes a long time. So it's better to prevent that by doing regular foot care. Another problem with feet that we have experienced several times in Sumatra is complete or partial destruction of the footbed in situations where elephants kept for a long time in dirty places, muddy places, wet places. In these places, a lot of bacteria develop, which are very aggressive and can destroy the footbed if you can see on this picture here, only the heel is destructed, but also larger parts of the footpad get destroyed if the elephants are forced to stay for a long time in dirty, muddy, polluted and wet places. Yeah, and this is after the first start of treatment, trimming down on the foot. Only part of the footpad is left. And the problem here is often that the tissue, the affected tissue, is granulating but doesn't perform proper epithelialization. So if that is not properly treated, these wounds are not really closing, and I have seen several elephants that died from this problem due to septicemia finally. And very often we can experience, or have experienced at least in the past, this problem in male elephants during the must, when they're not can be properly handled, safely handled, and they're chained up in one place and they're kept permanently in this place. In addition to that, it is if it is raining season, the situation becomes even worse, and very often these problems occur. So that is something we also have to pay attention as vets to try to interfere early enough. Another important part of our preventative schemes is regular antiparasitic treatments. Why do we need to do that? I mean, also wild elephants have parasites, but normally and relatively low or medium loads, which does not cause any clinical problems. But in captivity, elephants are restricted to smaller spaces where infestation with worm eggs are a lot faster, a lot more often, which can cause high infestations with gastrointestinal parasites, which can lead to severe sickness and, the worst case, even death. Therefore, also every 3 to 4 months, all of our elephants are regularly treated with different kinds of drugs in accordance with the worm species that we identify. So if you don't do that, you can end up with elephants which show these symptoms like weight loss, ventral edema in this massive ventral edema, loss of appetite, lethargy, diarrhea, sometimes even constipation from high worm loads. And in the blood analysis, we usually will find anemia, hypoproteinemia, and eosinophilia. Well, of course, these are symptoms that you also can see with other diseases. Finally, diagnosed, of course, is from the feces. Either you find the adult worms in the feces, then the infestations are very high, or you do a fecal examination with a microscope to identify not only that you have worms through the worm eggs, but also what species you have. So different kind of Strongyloides, Strongylus species, different kind of trematodes. Trematodes can be found in elephants, as well as cestodes. In Sumatra, the most often that we have different Strongylos, Strongyloides, and gastrointestinal trematodes from the group of the Amphistomum. Rarely we find fasciolosis in Sumatra. Very rarely we find cestodes, the Anoplocephala, and personally schistosoma have been reported in elephants from other areas in Asia, but I haven't seen it in Sumatran elephants. For us, a big issue in Sumatra very often is the Amphistomum. And amphistomiasis is caused by different Amphistomum species, Gastrodiscus, Pseudodiscus, Pfanderius, so different genera of Amphistomum can occur. And these are pictures from a post-mortem that actually died over after a long time of infection. And this is the pictures of the post-mortem. The entire gastrointestinal wall of the large intestine is completely covered with these worms. Yeah. So to prevent these diseases from high worm infestation, we use different kinds of drugs in the rolling scheme. We use different kinds of benzimidazoles, which mainly affect the nematoda. Some, like albendazole, also affect trematoda. We use the ivermectin, which is very effective against a large number of nematoda, as well as ectoparasites like Hematomyces elephantis. You can use it, or we use it sometimes orally, sometimes rectally, but also via injection, just to reach proper blood levels. If we use it oral or rectal, the dosage is higher than compared with the parenteral intramuscular injection. Yeah. The salicin analytes, we use closantel, which affects nematoda and also would affect fasciola. But the only one that affects, in our experience, where we had sufficient effect against Amphistomum, the amphistomin species that we find in Sumatran elephants, was the drug oxychlorosanide. Yeah, it also affects some nematoda and fasciola, but important for us is here that we have a drug that affects sufficiently the amphistomiasis, which is not done by closantel and is not done by any of the benzimidazoles we use. So these are the regular range of different dewormers that we use in a rolling scheme. We have also used a few others, like the benzimidazole triclabendazole, which is the choice of drug for fasciolosis because it affects all larvae from the fasciola, not only the adult, but it's effective only against fasciola. Praziquantel we have used, which is probably choice of drug for schistosoma and cestoda. As I said, schistosoma we have not found, but cestoda sometimes we find. And the imidazole levamisole is probably a choice of drugs for cutaneous filariosis. However, it also affects nematoda. The critical thing where I want to mention with levamisole, it has a very narrow safety margin. So only overdosing 1.5 times will cause very severe side effects, and only double the normal dose can, in the worst case, also kill the elephant. So I won't only recommend to use levamisole if you can do a very proper dose calculation, have a very accurate weight of your elephant. Finally, tetanus vaccination is one of the preventative schemes. Why do we do that? Well, tetanus is caused by Clostridium tetani, an anaerobe, obligate anaerobe bacterium, which cannot survive without the presence of oxygen, and is very heat sensitive. But it does produce spores, which you can see in the microscope, and these spores are very hardened, survive very long in the environment, and are more or less available everywhere in the soil. And if these spores, through, for example, a stepping wound with a foreign body, get inoculated into the body in a wound, deep in the wound where there's no oxygen, then the vegetative form starts to develop and it starts to produce toxin. And this toxin interferes with the neuronal signal transmission, which is causing spasm and muscular cramps, which mostly is leading to death. Well, tetanus infection is treatable, but in our experience, only if you start very early, when you catch the very early signs, which is usually lockjaw, so the throat of the elephant is already paralyzed, the elephant cannot swallow anymore, or partially paralysis of the trunk. Then very crucial is, and absolutely important is, administration of high doses of anti-tetanus serum, 200 to 300 international units twice a day. The challenges for us, at least here in Sumatra, is that is usually not immediately available in high quantities. Yeah? And of course, there is a potential risk of an allergic shock with this, especially if you may give it intravenously. I personally haven't experienced, but that's in principle is a risk. And it's very expensive, at least here in Indonesia, it's very expensive. If you have to do a treatment over a period of 10 days with 300 international units twice a day, we have to spend about 10,000 US dollars. Yeah. Apart from that, well, there's a picture of the anti-tetanus serum. If we can identify the wound, which is the source of the tetanus infection, of course, antiseptic and surgical wound management, but that's not always the case. It can also be an internal wound, like a gastrointestinal ulcer or something that is the source of the infection. Treatment with a high dose of antibiotics. And then as the elephant cannot swallow anymore, of course, we have to substitute fluid intravenously. And intravenously, we usually don't achieve enough to maintain the elephant, which is not able to drink now for a longer time. So also additional rectal fluids, and also forced feeding with a stomach tube can be indicated if the elephant is unable to swallow for several days or weeks, or sometimes even up to 2 months in the longest case that we have handled. Yeah, you can also do a sedation with the elephant to keep it calm in a quiet environment. And sometimes recommended is do slinging. But in my experience, if the elephant is collapsing, then it's anyway too late, because then your initiation of the anti-tetanus serum treatment has started too late, and the elephant will painfully die anyway. And in my opinion, then it is better to euthanize an animal. So to prevent that, we do in all the elephants that we are involved in the captive elephants, we do annual tetanus vaccinations, for which different papers are available that recommend 1 to a double horse dose in elephants, producing sufficient titers. Unfortunately, here in Indonesia, the tetanus toxoid vaccine specifically for horses is normally not available. There is no importer that makes it available regularly in Indonesia. So we have started to use several years ago already human vaccines, which are available from a company in Indonesia, which is also seen as available in other Southeast Asian countries. If it's the first vaccination, then this vaccination should be or needs to be repeated after 4 to 6 weeks after the first vaccination, after the first dose, and then is followed by a single dose once a year in each individual. So we use 5 milliliters. So the human dose of this vaccine is 0.5 ml. So we use 10 times the human dose. We haven't done an evaluation of the titers using this one, only with the horse vaccine. But since we're using that so far, at least we haven't had any cases in the population that we are involved in. So apart from captive elephants, we also get involved more or less regular in wild elephant situations, treatment of rescued and injured elephants, translocation of elephants, fitting GPS collars, and postmortems. So different kind of injuries in wild elephants which we try to treat in the wild, like a huge wound as a result from a gunshot wound, miasis in a wound on the vulva. I think this is an elephant. This was an elephant that has climbed several times over a barbed wire to get into the farmland of a local community and had an infected wound with miasis on the lower back. And this is an elephant that has been stabbed by spear trap, which is a very nasty part of a elephant trap that is used in has been used in some parts of North Sumatra. And of course, snare wounds. I think most people that work with wildlife know snare wounds. We also find that in the elephants, of course. If possible, we treat the elephants in the wild and let them go directly, but in some cases we have to take them into captivity to continue the care properly. Translocation of wild elephants, of course, is a very controversial issue. Nevertheless, sometimes the conservation agencies decide to do that because there are individuals or very small group which are isolated in farmland and are threatened to be killed, poisoned, or shot by the farmers. And then, of course, we also have to support or fitting GPS collars for wild elephant monitoring, for human-elephant conflict migration, and to understand the migration routes. We are involved in these operations. All of these operations, as you can easily imagine, require sedation or anesthesia, some form of anesthesia or sedation. In Indonesia, the tranquilizers or sedatives which are available are limited. 10% xylazine, 10% ketamine are usually always available. Solitile 50 and low percentage 2% xylazine and 0.1% medetim is available. However, these two drugs are not very suitable for elephants because the concentration is very low. That means we will need high volumes, which are too big to put in a dart. But so far, xylazine ketamine combinations have been proven to be sufficient for reliable standing sedations in wild and captive Sumatran elephants for different needs. And we also sometimes have high concentrated medetomidine, 30 to 40 milligram per milliliter available, which is imported. And with these high concentrations, of course, we can achieve very low volumina where we can use 2 to 3 ml darts, which of course is very nice. These are the dosages of xylazine ketamine combination, which you use in wild elephant. In captive elephants, if you use it in captive elephants, it's about 2/3 of these dosages, and often we need to top up this dose if we have to deepen the sedation, or if we have to extend the sedation. Of course, you can imagine a wild elephant, usually we don't know the weight, so we have to estimate the body weight. once the elephants are sedated, we also measure the elephants and then recalculate what was the actual dose. That is so how we know about which dosage we are given. So you see there's a very high, relatively high range, and depending on the effect or how deep the sedation is or how long the procedure is, sometimes some 30 to 50 minutes after the initial dose, we need to give a top up, which is about 1/3 to half of the initial amount. To reverse the xylazine sedation, we use usually yohimbine, 0.05 to 0.11 milligram per kilogram body weight, depending again on the procedure, about 45 to 9 minutes after the initial administration of the tranquilizer. In cases where we have high concentrated medetomidine, we use 6 to 8 microgram of medetomidine per kilogram body weight combined with 250 to 500 milligram solitile. So this is for wild elephants because the sedative effect takes faster, is faster initiated with medetomidine, but the half-life is shorter. So in case where we have to extend or prolong or deepen the sedation, then we use xylazine ketamine, because then we can inject it manually just by hand because the elephant is already partially sedated. Important is for the reversal of this medetomidine sedation, we have to use atipamezole. That yohimbine is not sufficiently effective, not sufficiently reversing medetomidine. The atipamezole is reversing both medetomidine and xylazine. Well, finally, just a very few words. Sometimes we also have to do postmortems in wild elephants, which always comes with a lot of challenges. As you probably can imagine, these elephants are found detected somewhere in the forest. The animals are very big. They cannot easily be removed, so you need a lot of heavy-duty equipment like knives, axes, saws, bolt cutters. You need a bunch of people to help you to cut the entire body apart and be able to take some samples and look inside. And of course, the entire logistical situation is often very difficult, like here in a riverbed, where you also have to watch out if it starts raining, if the river may get flooded, and not always you can get the elephant in a proper position. And of course, often also the carcass is already in decay, and the samples that you can take are limited. And also the possibility to store samples. We don't have electricity in there. We often don't have appropriate cooling facilities. So the results that you gain from these postmortems is very often not ideal and not perfect, but in each situation, you only can try what is possible with the given conditions and try to get the best result out of it for what you have done. Yeah, that a little bit in a nutshell is some things that we are working on in Sumatra and some of the experience that we have gathered in Sumatra with the work with the captive and with the wild elephants in Sumatra. I would really thank very much the organizations that support our work here in Sumatra for the elephant veterinary work, which is US Fish and Wildlife Service, the International Elephant Project, Asian Elephant Support, Durga Wildlife Fund, and Zoos Victoria. Thank you very much for your support, and I would like to thank you all for your attention. Thank you very much.
Yeah. Thank you, Dr. Christopher, for a wonderful presentation. It was very interesting and very illustrative also. Plenty of elephant. Let me check if there is any questions for you. I think there is one question. I will like to just read it out so that it will be recorded. Yes, there is one question from Dr. Azad Mohammed. And the question is, it's a nice presentation. I wanted to ask either you have any experience to treat an elephant in captivity with knee joint pain and swelling caused by long-standing time. Is there any specific solution or preventive measure to avoid this problem? Thank you. Yeah.
I don't have, so we have not a lot geriatric problems, often as a geriatric problem. If you have a problem problem like that, of course, you need to try to see if there is an underlying problem. Has the elephant probably some orthopedic problems? Is there some difference in the length? Because I know that has happened not in our place, but in other place, length with one of the feet. And of course, if it is an older elephant that has chronic arthritis, then of course, I think you only can do symptomatic treatment with pain medications. Another point is, of course, you have to look into the keeping condition. On what ground is the elephant kept? Is the elephant kept permanently on hard ground? Is there a possibility? Is the elephant? What is the work activities of the elephants? The daily activities? Is that something that contributes, or is it probably arthritis, age-related arthritis? Which then I don't think there is a lot of things that you can do apart from pain medication.
Yeah, thank you, Christopher. Another question is, when do we start the tetanus vaccination?
Well, we do start the tetanus vaccination. So if you have captive-born calves, if they are 8 months, 10 months, then we usually already do the first vaccination. Yeah, so below 1 year. So yeah. And with any other elephant, anytime. So if you have an adult elephant which is not in a vaccination scheme yet, yeah, you can start anytime, of course. Yeah.
Dr. Bhaskar has a question. It's a nice presentation. Purpose of management of captive elephants in Sumatra, for patrol in forest or tourism?
Well, the major reason why we have a captive elephant population dates back to the 80s and 90s, when the Indonesian government decided to capture elephants from the wild to reduce human-elephant conflict. Yeah, so there was not really the reason to take elephants from the wild, because there was a purpose to use the elephants for, but the Indonesian government for almost 2 decades had the policy, or had a part of the policy for human conflict mitigation, to just simply capture these elephants, which has not solved the problem, because the decline of habitat has continued. And initially there was not really a appropriate concept of what to do and how to utilize these elephants. Nowadays, a good number of these elephants which has survived is distributed to zoos, safari parks within Indonesia. So you can say basically the tourism, but also more and more during the past year, there has been common to utilize elephants in different, or on the border of different protected area and wild elephant habitats, to use them to do patrols and for human-elephant conflict mitigation, monitor wild elephants and push wild elephants back from the border of the farmland, push them back into forests. So that also is used in several areas, and I'm myself involved in programs where this strategy has been implemented or is implemented since several years. Yeah.
Okay. Dr. Ganga has a question. How common are the wildlife crimes involving elephants in Sumatra?
Relatively common. So we have basically, I would say, two levels. We have the illegal killing just to get rid of the elephants, if the elephants are considered pests by farmers or by plantation companies, yeah, where elephants get poisoned or electrocuted, or the wildlife crime where actually elephant poached just to gather parts, body parts, mainly ivory. Yeah, it is unfortunately also relatively common, and at least in some areas where we still have populations, the male population is quite significantly depleted due to this poaching.
Yeah, thank you so much. Dr. Ilayaraja Selvaraj has given some points about joint swelling. Just suggesting apply DMSO plus salicylic acid ointment, orally give trypsin plus bromelain, plus ceratolipopeptidase tablets. So this is his suggestion. So you have a few more questions. Sir, good presentation. This is from Dr. Selvam. Is it necessary to do TT at the time of wound treatment?
Well, if your elephant is not vaccinated yet, I would definitely consider that. Yeah. But we need to keep in mind that if we start with a tetanus toxoid vaccination, it will take at least 10 to 12 days until there is a sufficient antibody level. So if you have a wound which you are suspicious that may develop tetanus, I would go dual. I would give anti-tetanus serum for direct protection, and then start a vaccination. Yeah, start a vaccination of them. But if you need immediate protection, then the toxoid vaccine will not help you, because that will take at least 10 to 12 days until you have a titer. So then it is better to use the anti-tetanus serum. Yeah.
Yeah. Dr. Gurudutt Sharma has a question. Very informative session, sir. Which anti-helminthic drug you found most effective and safest for elephants, both for adults and calves?
Well, I can only say, and so far the ones that I have shown, whether it's albendazole, ivermectin, oxyclosanide, closantel, I haven't seen any problems in any of these antiparasitics, regardless whether it's adult elephants, old elephants, young elephants. And usually we start to deworm our elephants with an age of 6 months. Yeah. In the same scheme like the adult elephants. The only drug which I've mentioned, levamisole, where I have seen severe side effects if you accidentally overdose. So that I would be very careful with. For the others, I personally, which I have mentioned, all would consider fairly safe, and they have all a fairly wide safety margin. Yeah.
Okay, Dr. Yaduraj has a question. Nice presentation. Do you regularly use any other vaccines except tetanus? What dose?
No, we don't. We don't. Not because I would definitely consider in elephants. I would definitely consider rabies vaccination, because rabies is present in here. It's also reported that it has affected elephants we don't have reports in Sumatra, but from other areas in Asia. But that is a little bit sensitive in here, because it's not our elephants. We cannot decide. We have to apply to the Central Conservation Agency, come up with a proposal, and we have that done in the past. And for rabies vaccination, that has not been permitted.
Yeah, thank you.
Because with the explanation, or with the argument that there is no specific rabies vaccine specifically for elephants, which, in my opinion, doesn't make a lot of sense, but that's what was the explanation.
Yeah, thank you. Next question. No name is not given, but he has got 2 questions. One, are you practicing any other vaccination protocols in elephants like FMD, HS, and BQ?
No.
Yeah.
No, for the same reasons that I just also mentioned for rabies. We have considered that, we had discussed that, but in the process to suggest that to the government authorities which are in charge for the elephants, that has not been accepted yet.
Okay. The second question is, how about using pentosan polysulfate in chronic arthritis?
Well, I don't know. I have not used it. I don't have any severe problems with that with the elephants. I think that is a question that should be forwarded to colleagues that work with a population where a lot older elephants that have a lot of more joint problems. Unfortunately, I cannot give any good answer to that. I'm sorry.
Okay. Dr. Ilayaraja has a question. Do you encounter any cutaneous filariasis in your elephants?
We do, but not very often. Not very often. We have miasis, of course, wound miasis we have, which also then we always apply ivermectin parenterally. But cutaneous miasis only in a very, very single few cases. It's not a regular. We don't see that as a regular problem in the elephants.
Okay, this may be the last question for you, Dr. Ajay from Jharkhand. It was a very nice presentation. Is any incidents of anthrax you have encountered?
None of which we have proven anthrax. Throughout the past 20 years, there were 2 or 3 cases where I not was directly involved in the postmortem and in the sampling, where I think it could have been anthrax, but there is not a proven case that we say we have proven by laboratory findings that we had anthrax. But I definitely would never dismiss it. Yeah.
Yeah, thank you, Dr. Christopher Cremy, for all the replies. Now I just hand over the phone to just, Dr. Heidi. Yeah, thank you.
Great. Thank you very much, Christopher, and thank you very much, Daniel. So we will move on to the next presentation. The next presenter is Dr. Arun Zakaria. Dr. Zakaria is a Chief Forest Veterinary Officer from Kerala State in India. Since 1994, Dr. Zakaria has been working as a wildlife veterinarian for the government of Kerala with both the Department of Animal Husbandry as well as the Department of Forest and Wildlife. Dr. Zakaria is experienced in disease surveillance and epidemiological studies in free-ranging wildlife, as well as in laboratory techniques that aid in disease diagnosis, including molecular diagnostic methods in infectious disease of veterinary importance. Dr. Zakaria's presentation will be about disease ecology and emerging diseases in free-ranging Asian elephants. So, Dr. Arun?
Thank you, Heidi.
Please start whenever.
Am I audible?
Yes, you are.
Okay, let me share. So, okay, can you see the slide?
Yes.
Okay, so thank you very much. Thanks a lot meeting all and sharing some of our experiences. And okay, so this is the topic given to me, disease ecology and emerging diseases in free-ranging Asian elephants. So I'm working as a forest veterinary officer with the forest department for the last 22 years. So we just want to sum up some of our findings and what you have seen in the field. So basically, I'm working with free-ranging wildlife. So whatever we are covering with the free-ranging wildlife mostly. Okay, so looking to our topic, we got around 350 species of proboscideans. Only two genera now survives or exist. Rest of them are extant. Like you can have Dinotheriums, Mastodons, Gomphotherium, and so many of them became extinct. And looked like in Paleoloxodon, the huge elephants to pygmy mammoths. So what would be the reason for this extinction, this mass herbivore extinctions? So one hypothesis that put early, 50 years back, was a hyperdisease concept. Okay, so hyperdisease is just like a pandemic really causes extinction. And one of the examples given by Rothschild and Loeb in 2006, they identified lots of MTB lesions or tuberculosis lesion in the fossils of mastodons, especially American mastodons. So they said, okay, TB is inverted through human migration into North America or all across America and caused the extinctions of many herbivores, especially American mastodons. So this is quite interesting. Actually, it's not nobody noticed this paper much, but this gives an indication. Of course, disease has a role in cause of extinction in many of the species, especially larger herbivores. Okay, so as you know, in the Asian elephants, and this is the distribution of Asian elephants, you can see mainly seen in 13 countries and the largest population in India. So current problems in elephant conservations it's mainly due to high density, due to habitat degradation and fragmentation, and that compromise with the home range requirement. So actually this pushes the elephants into, in thick groups, and which create a lot of issues. And second, due to extensive large poaching and this habitat fragmentation, genetic variability is coming down in elephants in many populations. And there is a high level of man-elephant conflict, as Christopher told, especially in Sumatra and part of India also, and which threats the conservation. And there is a highly skewed male-female ratios. And in some of the places in India, the male-female ratio is 1:200, and which impairs lots of selection process, including sexual selection process. Then there is a cattle grazing problem. The landscape being used by so many other purposes, including agriculture, animal husbandry practices. And of course, emerging infectious diseases are identified as a new threat. And the real problem is actually we don't have many previous research on mortality or morbidity due to diseases in elephants. Hardly, I couldn't say 1 or 2 PhD theses and very limited papers. Looking into the paper number, I would say 98% of papers are based upon the captive elephants. Only 2% of papers are based on free-ranging wildlife. So very limited data is available. So Asian elephants are quite unique, the longevity, just like a human being. Most of them live up to 70 years and up to 80 in captivity, and mostly in 60 to 70 if they are survive in the wild. And puberty and age of sexual maturity is almost just like in human, 13, 14 years, they become in 10, 12, they become attain the puberty, and by 13 they attain the age of sexual maturity, just like in human being. And the social organization is quite important. It's a matriarchal society, the lead female, and she decides the survival of the herd. And the sexual dimorphism and the sexual selection, which is quite interesting. Not like in African counterpart, we have a sexual dimorphism. The bulls have tusk and females have, most of the time, they don't have tusk, or they may have small tushes. And there's a huge sexual selection is happening, okay, which is quite important in Asian elephants because females are selecting best bulls. There will be a male-to-male competition during the must season and female selecting because she's investing the energy. And the behavior is quite interesting in elephants. There's a changing in behavior pattern looking into human-wildlife conflict. Now we are facing with large number of huge bull congregations creating conflict, and the natality and mortality. Okay. So the problem with us working in free-ranging wildlife, especially in elephants, how would we monitor the health in the wild elephants, and how we could say how fit or how healthier our wild elephants are? First, we can look into body index, look into the body. There are body index parameters. There are a lot of references you can look into. And we can look into the body index and we can say, okay, this body index is very low. And we can say the herd is not healthy or our population is not healthy. If it is in the captive situation, always you can have a clinical examination like Christopher told, you can go for pulse, respiration, temperature, blood picture, serum biochemistry, everything to look into. And you can come to a condition of the health status. And in the free ranging, you can go for non-invasive sampling also. You can collect the dung and urine sample if you are lucky enough to get it fresh. And look into the disease conditions in those samples and come to an epidemiological picture. Okay, then finally, and looking into morbidity and mortality in the population and to identify the health in the wild elephants, and which is a more practical, this approach we are doing postmortems. So far we did more than 620 postmortems in elephants. And so we will get a clean picture of what's really happening because you're looking, every postmortem is quite important. You're opening a carcass and you're seeing the real lesions if you're lucky enough, if it is not putrefied. Okay, looking at the body index. See, this is a situation in summer in my landscape. See the body condition. You can see the dipping of shoulders. You can see the ribs. And all indexes are very negative. And you can say this population is unhealthy. You can imagine. But when the rain comes, the whole situation changes. So body index cannot be used in most of the situations because if the population depends on seasons availability, elephants tend to change browse and grays depend on the season. If it is an evergreen, okay, you can count on body index. But if the seasonal changes happens, elephants tend to browse and graze changes, and definitely there will be changes in body indexes, and which cannot be used as a legitimate index to identify the health condition. Okay, then regarding the non-sampling, I'm not talking about, I mean, the captive situation. So non-invasive sampling, as I told, urine, or you can get fecal sample fresh from the wild. So the problem is that many of the time, the pathogens are not shed through either feces or urine. Okay, and sometimes especially like viruses are very fragile. Okay, we try to identify herpes virus in fecal samples, and our success rate is very, very low. But we know that the animal is affected with the herpes virus simply because the virus is not stable at all. So it may not work in most of highly fragile pathogen, especially viruses. Then good for assessing prevalence of internal parasite. Definitely you can use it. That is the best way to do it. And problem is that difficult to correlate with the interpret epidemiological data derived, because if you are not identifying the population size and age structure, you cannot come into an epidemiological data. To identify the age and sex from the non-invasive sample is very difficult and time consuming and costly as well. So it's very difficult to correlate to come into an epidemiological data. And finally, it left with the mortality in Asian elephant. This is not a clean, good this thing. The mortality in Asian elephants, it fluctuates. It's just not like in a single plethora, like in the human being. In the human beings, you can expect a percentage of mortality in some seasons. Okay, but it's almost like a study. But when you look into elephants, it fluctuates. Every 4 years, you are getting a fluctuations of mortality, and which is quite interesting. And there are so many variables you can correlate, especially the climate change also you can correlate. And season-wise also, especially I told you, elephants which are depending on the seasons, there will be more changes of mortality during the season. For example, in our population, we are having high mortality during summer season, another peak during September, October, November, December season. And those two also, when you correlate, you can find the summer mortality is much more to old elephants and young elephants. And the October, November mortality is mainly in prime bulls, mainly due to fight or other kind of breeding problems. Okay, so there are a lot of dilemma in veterinary profession in care of free-ranging wildlife. Okay, the problem is, should we have to look into individual or into populations? As I told you, when you look into the papers, you can find more of the case reports rather than epidemiological population data, which is quite, you cannot interpret those things. So should we have to focus on individual, or should we have to focus on the population wise? And second question is, should we have to focus on welfare or the conservation? Most of the concept is based upon the welfare, because most of the science derived from captive elephants, which is based on welfare. So people used to confuse, veterinarians used to confuse, should we have to focus on welfare or go to conservation? And third importance, the unique evolution of Asian elephants is an Afrothetan species. Okay, it's actually evolved in northeastern Africa and came to Asia. Okay, so their evolutionary thing is entirely different. And most of the pathogens, especially the internal parasites, are unique for elephants. You cannot find much of parasites from other species, just like you cannot find internal parasite of a gore or Sambadia in elephants. Then the fourth one, again, I told the lack of knowledge or information of disease ecology of free-ranging wild elephants, so wild animals, basically. So we are clearly deprived of data. So conceptual inabilities. This is another problem. So people used to, especially veterinarians who trained, because we are not trained in wildlife, in captive situations, we think disease is a negative point, but most of the conditions, I would say, in the population wilds or in the free-ranging situation, disease as a major natural selection process. I will explain that later. Then coming to that, defining the level of intervention, where and when to be intervened. Should we have to intervene? That is a question in a population. Okay, if there is a death due to a herpes virus, should we have to intervene? Should we have to vaccinate all the animals? And those kind of questions. So I used to get questions from the people asking, why can't we develop oral vaccines for many diseases like that? So the question is, should we have to intervene? That's a dilemma. So these are the main dilemma what we are facing as a veterinarian in the case of free-ranging wild elephants. And coming to an example of this thing, parasitism versus predation. Okay, so this is the right one. You can find an elephant calf killed by a tiger. You know, the tiger is a major predator of elephants, especially in the young animals, not an adult one. So we analyzed around 132 cases of these kind of predations, and we found that all of them, 92% of them, had infected huge parasitic load. Okay, so it is called parasitic mediated predation. This parasite is actually mediated predation. So this is a selection process is happening. Parasite mediates that reduces the fitness of the animal, which lead to predation, and the selection process happening. So kind of that conceptual problems exist. So when this all coming to in the free-ranging situation, we have to consider the disease in a multi-dimensional view. You have to look into an agent as a host and the environment, and not like in a captive situation where we may mainly look into the agent. What is the disease really causing? What is the pathogen really inflicting the disease? But in a free-ranging situation, you have to look into the agent, the host, maybe the genetics, immunity, and other ecological parameters. And the third is the environment, which is quite important nowadays. Okay, so my presentation focus on these 3 points. Okay, so the changes in any of these will change the triad and the disease epidemiology. Okay, so that gives us the concept of One Health. Okay, so looking into a free-ranging elephant situation, always we should have a wildlife disease ecology. So wildlife disease ecology is simply defined how do climate, evolution, and free living hosts interact to determine the dynamics of pathogen and burden of diseases. So Hudson actually defined it in 2002. It's a wildlife disease ecology defined as an ecological study of host pathogen interaction within the context of the environment, as a triad I mentioned earlier. So it's better to have a disease ecology aspect. The wildlife disease ecology is something a new science, very nascent science. And looking into wildlife disease situation always should have a disease ecological perspective. Okay, so I will just simply explain some of the mortality, cause of mortality. So I told you the predation, natural causes, which are happening naturally in a population. Sometimes the elephant can fall, right? So our predation rate is around 9 to 10% in elephants, because the young animals are killed by tigers, mainly by tigers. Then sometimes they can fall, they can slip and fall. And for example, in this case, you can find a healthy animal. You cannot find much problems, no fractures, nothing, but just the animal is stripped with a small vine and it broke, it just gone into the mud and it broke its atlanto-occipital joint. So animal can fall and die. And there are other causes. They can fight, huge fight incidents happening. Okay, our bull mortality rate is around 10%. Okay, in the largest global population, that is the NBR, Nilgiri bear sort of population, they can fight male-to-male competition, especially during November, December, when there is a musk condition. They come, they do fight, and they kill each other. And for example, in this case, it's quite interesting. That has happened in Periyar Tiger Reserve 10 years back. A single bull killed 22 females. Okay, usually it's very quite rare, bulls killing females. So these aberrant things can also happen. Then coming to anthropogenic reasons, man-made issues, mainly killing for ivory, luckily which is came down quite less because of intense activity of the forest department. And sometimes due to natural electrocutions. Okay, lightning can cause. The left one is a lightning case. It killed, wiped out the whole herd. And right side is an electrocution due to a man-made. There is a high power line. Sometimes some are due, they may slag. So they may touch and they can kill. So there is both natural and by accidents. And these are electrocution accidents. People used to put electric lines for bushmeat, for killing wild boars or deers. Sometimes elephant got electrocuted in that. Sometimes they do poisoning. This is mainly to mitigate human-wildlife conflict. That's exactly Christopher told earlier. So sometimes if it is an animal come to conflict, they do poison. Okay, they dies. Sometimes they put country bombs, that explosives hidden in a jackfruit or maybe pineapple, so can cause death. And there are other non-infectious diseases as well, like parasitic diseases, which can be infectious and endemic also. But at the same time, we find a lot of cases of peritonitis, okay, in elephants, mainly due to penetration wounds, especially bullets or other kind of things. So looking into disease, and we can basically classify based upon etiology and or epidemiological pattern. Based upon etiology, you can classify as microparasites and macroparasites. Okay, and the epidemiological, microparasites are just viruses. You can see them, bacteria, all kind of things. And macroparasites are mainly helminths, trematodes, those kind of things, cestodes. And epidemiologically, you can define them. There are some diseases inherent to diseases that are diseases evolved along with the elephants. I will give you some examples. And so there are some other diseases affecting multiple taxa. The dynamics is different. They can affect a goat, a boar, sometimes a cattle. Okay, and comes the emerging diseases. For example, in this interesting case, there's a stomach of an elephant. You can find boards, okay, in the stomach, which is very common. 90% of elephants, you can find these boards. These are boards of a larvae of a fly called Koboldi elephantine. Okay, they lay eggs on the commissures of the mouth or maybe around the tusk, and elephant swallow it. It hatch in the stomach, and larvae pupate, and the pupa breaks, and the fly through the mouth. Okay, this is the only place in the world this particular fly can survive in the world. Okay, only in the elephant stomach. Okay, this is called coevolution. This fly is evolved with the elephants. Okay, the elephant dies, fly gone. So there are some diseases which evolved along with the elephants. For example, another one, in the case of this is an elephant calf. This around 60% of our elephant calves dying because of it's called Grammocephalus species viridatus or Grammocephalus species viridatus, and which are large hookworms affecting liver. This only pathogen you can find. This is a major 60% of mortality in elephant calves because of it in the free-range wildlife. So, but they can kill the animal also. Okay, this is what is called density-dependent selection. Okay, when the population increases, these parasites actually controlling the population, and this is one of the major selection process. Okay, so all right. Okay, this is the case of an anthrax. I would say there are some diseases which can affect multiple taxa. So they can kill an elephant, and they can kill the wild boar. Right, they say wild pigs are resistant with anthrax. No, there are lots of anthrax cases in wild boar. They can kill. So they're basically from the soil. The spores they're getting the soil. Their dynamics are entirely different. Then coming to emerging infectious diseases, so which is quite interesting because we've identified a lot of emerging infectious diseases coming up in Asian elephants. So this is a definition by the Moores, those diseases which have appeared in a population for the first time, or those diseases which have existed but are rapidly increasing the prevalence or geological area. So there are lots of emerging infectious diseases. There are wildlife EIDs, there are human EIDs, there are domestic EIDs, which intervening, okay, which are connected. Okay, they are called interface. There are interfaces. They are connecting, and there is a transmission of emerging infectious diseases like hantaviruses in humans or equines. They're getting from the bats. Okay, these are spillover events. Otherwise called spillover. There are so many drivers for that. Global travel, human encroachment, translocation, environmental changes, agriculture, the same practices, technology practices. There are so many drivers for them, but these are the major EIDs which can be classified separately. So we are working in the wildlife EID. For example, in the case of this an elephant killed by a parasite called Parabronema smithii, a small worm, which affects the young animals, causing tumors in the stomach, and subadult animals causing nodules in the intestine, which prevents them absorption. Animal dies. And usually we didn't get much of the cases. Now the prevalence is very high. So primarily we can say, I'm not saying it's a new disease, but I would say the prevalence is increasing, which can consider it as a emerging disease. So primarily, we think it is mainly due to congregation of elephants due to habitat loss. They push to in some places, so the transmission, the orophycal contamination is the route of transmission, and they get infections through that way. Then coming to spillover, the tuberculosis in elephants, this is a major problem in captive elephants, especially in European and American zoos, and definitely in captive situation in Asian conditions also, but not much in studies gone through it. So elephants are very prone to Mycobacterium tuberculosis, MTB. MTB is a human TB, evolved in the humans. Okay, as I told you, the hyper disease concept, which kills the mastodon, hypothetically kill the mastodon. Like the same thing happening. They get the infection from human beings. MTB is a human pathogen. Okay, and there are lots of these things. So we did a lot of postmortems. Then we got around 8 cases of MTB. So first, we are getting any kind of tubercle lesions. I will show you that. We do the impression smear and do the staining, acid-fast staining. We do the culture, very difficult to culture. We do the histopathology, but best thing is molecular methods, PCR and sequencing analysis for the species identification and typing. Okay, this is our first case of index in 2007. A young bull around 20 years in wild, okay, died. You can find the body condition is very poor, and these are the massive lesions. You can see that these are left side is the mesenteric lymph nodes, and right side you can find the tubercle. Okay, right. The same thing happening in the mediastinal lymph node, large lymph node with mucopurulin, and the right side is the lung condition, massively infected with the mucopurulin changes. When you do the histopathology, you can find clear tubercle nodules, right? Then you can identify the TB in acid-fast, histopathology, acid-fast, you can find impression smears also. So our cases, the mortality percentage is 6%, which is old data. Right now we have 8. Then MTB 4.7%. Only males affected, which is also wrong. We got recently a female case, and age group between 15 and 40 years. Okay, young animals are not being affected, but mainly adult animals. So significance of conservation. So we think this is so the first we got out of the 5 cases, we got it from bull elephants, which did the conflict. Okay, we thought so they pick up the infection. Otherwise, elephants, nobody can sneeze into, the transmission of TB is very difficult. It should be a 10 micron like droplets has to transmit it. Okay, if I get it, if I should get a TB patient should sneeze and I should inhale that droplet. But it's not possible. So a TB patient can catch hold of a wild elephant's trunk and sneeze into it. But the problem with that, first we identified with the captive elephants, but now we get a female elephants that is indicating it is circulating in the wild. Okay, so because females are not conflicting, very rarely conflicting in our landscape. So it is circulating in the herd, which is not a good news for us. Then pathogen spillover from the human being was the cause of emergence of Mycobacterium tuberculosis. So behavior may have a role because coming to human settlements, tourism, especially may be predisposing the conditions. Then coming to emergence of viral diseases, the EHV, I don't want to talk about much about it, the history of EHV. So we reported EHV first in 2007, the first case from the wild. Then we got around 46 cases of TB so far. And these are our captive TB cases. All of them are orphan calves. We put them in captivity because they are orphan. And many of them dies. Sometimes they transmit. And these are some of the wild cases. This old data, actually 22 cases, more than that now. So the middle one was the Intex case. Mother was guarding. The calf is alive, but died within a few hours. Okay, so I just want to show some of the lesions because you know it better. So, but that's a peracute onset. There will be temperature, anorexia, dullness, and facial edema, cyanosis of the tongue, and there is extensive pathological lesions, extensive hemorrhagic lesions, and this is the facial edema. You can see. Okay. You can see around the periorbital area, there should be edema, facial edema is there. And the tongue would be very bluish in color. First, you can find the salivation, then the tongue would be very bluish in color, clear cyanosis. And when you do the pathology, you can massive hemorrhagic lesions, just like epicardial lesions, massive lesions, and massive endocardial lesions involves, and the peritoneal lesions as well. Then the liver would be just like a balloon, okay, and you can appreciate the subcapsular lesions also, hemorrhages also. And you can find the lesions in the brain. There will be dullness and behavioral changes as well. So you can find mainly you can find the inclusion bodies in the liver or heart. But the best way to do is PCR. And these are the target genes we are using. So these are some PCR. And the story, actually, the herpes virus, as you know very well, first it thought as a species gem from African elephant to Asian elephants in zoos, but we identified first gem in wild. It was surprising. Then basically it's a beta herpes virus forming a new proboscis group. Okay, so it's finally, when we did the sequencing, we did around this 46 cases of sequencing, and we identified that from our landscape is also, it's actually co-evolved with the species. Okay, so the transmission is mainly through individual to individual close contact, mainly through secretions, trunk wash, because the main identification method is trunk wash. Adults are carriers. Okay, so these are cases we identified in the wild. We got the first case and we tracked that herd for almost 3 months on foot, and we got a second animal. Then we did the sequencing, whole viral sequencing, we exactly the same virus. Okay, so the molecular epidemiology is interesting. We added 31 different EHV1A genotype. Okay, I would say strain. I can't say strain, but genotype. Subtypes were identified, which is quite variable. Each population have its own genetic genotype. So Christoph has a different genotype, and Assam, they have a different genotype, definitely. When you look into hypervariable regions, it depends upon which region looking into. So go for very hypervariable regions, you can identify. There are 13 novel strains were identified. And quite interestingly, and there is no spatial or temporal epidemiological correlations, but sometimes they are much more seen during winter season, not winter for us, cold season, November, December, January. This is season. This month itself, I got 5 cases of herpes virus. So, but mainly during the weaning season, when they wean, so we are getting more cases in that group. So that age group of 4 to 5 years at the time of weaning, we are getting more herpes, not in the young calves. Young calves we are getting, when we are getting the orphan calves, we are very young animals, we are getting herpes viruses. But in the wild, when you look into those cases, you can find most of them are juveniles, which are undergoing the process of weaning. So, yeah, so the transmissions. Then viral shedding. So as you know, our first work we did identified viral shedding in adult animals. It's the same work what they did in US. We did that, and we identified so many of animals adult animals are shedding. They won't have any kind of clinical symptoms. So we tried the same thing in the wild. So we identified so many elephants. Actually, 2 elephants shed. That is wrong. Actually, there are 6 elephants so far we identified. When this opportunistic samples, when we did any kind of radio collaring operations, we collect the samples. We identified many females and bulls shedding viruses. So literally, I would say most of the elephant calves are born are under the shower of herpes virus in the natural situations. Okay, so why is some of that dying, but some of that not dying is quite interesting. So on the context, there are so many hypotheses. One group is saying it's due to colostrum deprivation, like I said previously. Some group is say, the herd transmissions when the herd moves in, and likewise. And the group like we say there are host genotype actually deciding the mortality. So on the context of this, whether we can say it's a truly emerging or co-evolved with the species? Definitely EHV is co-evolved. It's not a true emergence. But the problem is that the intensity of the cases is really increasing. Okay, because our predecessors, Krishnamurthy, he did a lot of postmortems. He got only 1 case, but now we are getting a lot of cases. In 2007, we started. Now we got cases from whole across, Dr. Bhaskar knows, whole across India. We got cases from Myanmar. then Indonesia, then Sri Lanka. So wherever there are natural population, we are identifying herpes virus. So why it is increasing, prevalence is increasing. There should be biases. We are looking into herpes virus. That's why there is one argument. There's one argument, but I cannot agree with that, because we are doing intensively postmortems, and why there is a real increase in spatial and temporal increase in incidence of herpes viruses. In that context, we can say it's emerging. Okay, we need more data to prove that. But whatever the impact, if it is emerging in such a situation, this is it's a survival 1x curve, mainly in elephants, just like in humans. It says that animals die as age goes on. Okay, again, animals are not prone to death easily, but as age goes on, animal dies. So it's affecting mainly the early the curve, and which can skew the curve and really causing the extinctions. So like small populations like in Indonesia and other part, we are quite worrying. It can really cause extinctions. If it is a meta population, we are facing same thing in some place in Munnar. So one of my colleagues is there, he knows this better. So that area, there are a lot of cases identifying in that small population. Okay, so it's a meta population. There is a chance of extinction of those meta populations, not a large scale extinction. The place where I'm working, the NBR, around 9,000 elephants. So, but can cause extinction in isolated small populations. Okay, this is the hypothesis we want to put forward. EHV1 have significant role in the Asian elephant population, could be emerging in the population. The loss of genetic variability, that is exactly we are focusing, like including the MHC1 variability, may have a direct relationship to the emergence of the disease. So this is sometimes a host genotype, what we are hypothesizing, may have a role. Okay, when there is an inbreeding happening, there is a high chance for the emergence. So since most of the herpes virus have latent infections or environment changes, and the stress in the population might have a direct relation to the emergence. Now we are coming to the environmental changes. So first I talk about a spillover, a pathogen change from the TB. Then I said the host changes, genotype changes. And now I would say a change in the environment. This is a case actually, we identified per acute mortality in 2014 to 2017. This is a small landscape number, but sudden death. Animal is dying just like a dropped apple. So only adult animals, no female, no male bias is there. Dead due to summer seasons, and grossly very difficult to identify from anthrax. They bloats like anything. Okay, within hours, they bloats like anything. And no other significant mortality in cohabiting species, just like other herbivores, just like deers or bison. Okay, then finally we got one case, fresh case. The mother is alive, well, mother is dying, and the calf is alive. And we got the fresh samples, and these are the pathological lesions, just like anthrax. In the beginning time, we were really hesitant because we got anthracoid-like organisms from the blood oozing out from the rectum, natural orifices. Then we did the spore PCR of anthrax, but it was negative. So we confidentially start doing the postmortems. And it just like the pathological lesions are, they're just like, typical anthrax-like lesions. So we try to culture that, but it's anaerobic. Anyway, we are very sure it's an anaerobic condition. So a culture was very difficult because in the anaerobic situation, there are a lot of other anaerobes flares up, and which will prevent, you cannot come into any kind of conclusions. So we applied whole genome sequencing, okay, bacterial metagenome sequencing, maybe the first time in free-ranging wildlife. So we use the Illumina platform to identify V3, V4 regions, amplify using specific primers. And when we did that, we got a huge bunch of cases and coming into this taxonomic group. Okay, Clostridium novae type B. Okay, then we develop Sanger specific sequences. Okay, we'll come to that. This is our NGS metagenome data. Then we validated with the 16S ribosomal RNA gene data of Clostridium novae, which is quite same. Then we developed, we use flagellin genes specific to Clostridium novae type B to type it, and it was very clear. So molecular findings are taxonomy annotation based upon 16S RNA, and Illumina data, or that is metagenome data, back to metagenome data, are very similar to the phylum Firmicutes and Clostridium novae. Then species level Clostridium novae was commonly present among all the samples, and the Shannon index rank, it was high. And further, Sanger validation due to 16S and also flagellin gene also confirmed Clostridium novae type B. Okay, Clostridium novae is quite important. It's very rarely described in wild animals, mainly in sheep. It's described black disease, except some cases in reindeers and bighorn sheep in Canada. Okay, so it's a truly emerging. Now every summer we are getting cases. Okay, it's again fluctuating as well. So there are other cases also. This is a quite interesting case. Around 6 cases. Dr. Kalivanan got a couple of cases. This is a systemic mycosis. Okay, just like look at the liver on the left bottom panel. You can find TB-like lesions, but the whole liver is filled with fungal mycosis. Okay, so this is a systemic mycosis, which quite rare. You can find the typical vulval lesions also, and could be attributed to the host immunity. Okay, systemic mycosis is very rare in a natural population, except some like patients, just like immunocompromised patients. In the humans, you are getting a lot of cases. So this is something we can looking into buccal and vulval mucous membrane examination. It can give any clues in immune status, because you know the galt and malt gut-associated lymphoid tissues and mucus-associated lymphoid tissues, which is very important in elephants' immunology. So they are first defense of innate immunity, and which can give kind of we are arguing kind of status of immune status. So thank you very much. So thanks a lot.
Thank you, Dr. Arun, for the wonderful presentation. It was very, very informative.
Sorry, I took more time. I'm really sorry.
No, that's fine. That's fine. No problem. Yeah, you have a few questions also, maybe 1 or 2 questions.
Yes.
So, can you brief and throw some light on herpes vaccination, please? Name is not given.
There is a new vaccine. There is a new vaccine developed. Okay. The group from Netherlands, they developed a vaccine, and the efficacy under tests, and I think it will coming out. It is under the process. I think it's under the process. So subunit vaccine. Okay. So there is a development of chance of vaccine is really promising.
Okay. Dr. Bhaskar has a question. Do you think rate of alteration of environment by anthropogenic factors also evoking some adaptations in the population of elephants? Further, smaller and fragmented populations will be more affected with such alterations. Are we seeing, a few more things. Are we seeing more disease in small and fragmented populations than in large populations in well-connected habitats? These are the questions.
Okay. Of course, yes. The question for the first one, that is large population versus small populations. So meta-populations, I would say, is if it is due to, human reasons, anthropogenic reasons, or natural reasons, which the meta-population problem is that actually it increases the contact area. Okay. When a large population, there is a large landscape, the chance of interaction with human to wildlife or interface is less. Okay, that is one, the spatial parameter. Second one, the genetic parameters. In a small population, the genetic fitness goes down. There is a high chance of inbreeding. As I told you, it's a female selection. So there is a less chance of selection process. And the genetic fitness of the population may decrease, which can impair, which can make this animal, okay, this is again a hypothesis based, but there are a lot of evidence coming up, causes the emergence of diseases. Yes. And the third question is, I'm sorry.
Are we seeing more disease in small and fragmented populations than
Yes,
large populations in well-connected habitats?
yes. The problem with that, when there's a large population, of course, the number is more. Okay, so incidence number is more. Okay, the frequency of disease is more. Okay, in a small population, so based upon the population size only, you can say, epidemiologically only you can say. Okay, so cases divided by the population size. Okay, so there are problems. There are new new recruits, just like birth. You have to look into natality and mortality, and coming into a population size. I would say an effective population size genetically, and then you have to correlate with that to the mortality. Then only you can come into that. Yes, that gives some indication that fragmented small populations, there is a high chance for diseases.
Okay. There is one more question from Mohammad Firdaus. He says, insightful presentation, Dr. Arun. I would like to ask, with regards to the systemic mycosis, what fungi was cultured from the case? Is it sporotrichosis or mucus? Yeah.
It's a sporotrichosis, actually. We didn't culture that. We did a PCR. Okay, it's a sporotrichosis, all the cases. So which is quite bizarre for us. Okay, so first we did the histopathology, we got this thing, and we did the PCR. So yeah, it's quite bizarre, and I think it's emerging conditions. We need more studies on that.
Okay. Yeah, thank you. Dr. Ganga from Sri Lanka has a question. Is it legal to euthanize critically ill wild elephants in India?
No. But we are trying to, change the laws, just like fractures or other cases, and we better to do the euthanasia rather than animal is suffering. So, but it is not legal. You can do that actually if the chief wildlife warden permits. Okay, the head of the forest department permits, we can do that most of the time. Yes, it's legal. Sorry, it's legal. It's legal, but it's not under the purview of a veterinarian. Okay, the domestic animals is under the purview of veterinarian, but in states, the chief wildlife. Sorry.
No, nothing. It's an auto message. Yeah.
Okay.
Yeah,
okay. Yeah. Okay. So so it's basically legal, chief wildlife warden, but it's not under the purview of a veterinarian. So we want to change that. We want to make it more flexible, right?
Okay, yeah. There is one question from Punika. Thank you for the interesting presentation. Did you find the incidence of hemorrhagic septicemia?
Not much, which is quite an interesting question. Not much. We have seen so many papers talking about hemorrhagic septicemia, but there are a couple of references from captive elephants, hemorrhagic septicemia, but not in the wild. We are extensively looking into hemorrhagic septicemia, but not in the wild.
Yeah. More questions may come. Thank you so much, Dr. An. Over to Dr. Heidi. Yeah.
Okay, thank you very much, Arun.
Thank you, Heidi.
Really appreciate your presentation.
Thank you.
Very interesting. So the next presentation, it's actually going to be a recorded presentation by Dr. Amir Sadaullah, who is in the field right now, and so his internet connection is not very good. Dr. Sadaullah is a wildlife veterinarian working with the National Trust for Nature Conservation in Nepal. He's been based in Chitwan National Park since 2014, and Dr. Sadaullah is involved in wildlife rescue and rehabilitation, captive elephant treatment, and wildlife disease research. So we will show the recorded presentation, and then Dr. Sadaullah will try to be online to answer your questions. So we'll run the presentation right now, the recorded presentation.
Good afternoon and namaste. Myself, Dr. Amir Sadaullah. I'm working as veterinary officer in National Trust for Nature Conservation Biodiversity Conservation Center. This series of webinar organized by Asian Elephant Support in partnership with Live Trust India. Today, as an episode which is organized about the elephant health, I'm presenting about the captive elephant health status in Nepal and the overall scenario we are getting across over here. Telling about the demography of captive elephant in Nepal, we have in government owned, we have around 104 captive elephant. Among them, 57 are based in Chitwan National Park. Similarly, 12 elephant are there in Bardia National Park. In Banker National Park, we have 6 elephant. In Parsa National Park, nearby to the Chitwan National Park, we have 9 captive elephants. Similarly, in Shukla Phata, we have 9 elephant. In Kosi Tapu, we have wildlife reserve, which is in the eastern part of Nepal. There is 11 captive elephant. These are all all governmentally owned elephant. And the interesting thing is, like if you look about the demographic data of few years back and now, we in all of this park now we have the young calf. They are each of them has capacity to breed and breeding. They have a breeding cow, and every this protected area we have young calf born in our captive setup. Similarly, in privately owned, we have 70 elephant. All of these are based in and around Chitwan National Park, and all of these are owned by the privately owned hotels who who like among them now we have now these elephant also have capability. They are some of them are also breeding and giving birth to the young calf. Similarly, if we talk about the use of the elephant in Nepal, generally the elephant are used for the park patrolling, like they are used for the used for patrolling, like for are used by the army for like anti-poaching patrolling. Similarly, they are some of these elephant they are used for the research activity, like to put on a camera to for a rhino census and all of these things. These elephant are used. Similarly, elephant has very important role in wildlife rescue and treatment. Similar when we go for the rescue of the tiger in very unknown area inside the park or in the forest, we are not able to perform the work without the presence of the elephant. These are the very important thing we need during a rescue. In this 2 photo, you could see the use of the captive elephant for the treatment of the rhino. Similarly, the treatment of the like rescue of the tiger, and similarly, these most of the governmentally owned elephant are used for the park patrolling, research activity, and rescue. Similarly, the elephant owned by the private owners, they are basically used for the ecotourism activity, like rescuing, like for the safari to the forest where the vehicle could not reach over there. Now I want to talk about the major health problem which which we encounter in our basic major health problem we encounter in our elephant in Nepal. The tuberculosis is one of the most important and important disease which affect our elephant, and this account for most death of the captive elephant in Nepal recently and in past as well. If you look on the history of the tuberculosis, the first case of the tuberculosis was reported in the patrolling bull in 2002, and we thought that this is the exceptional case, and then we got second case in 2004. When we get the 2 case regularly, it's an alarming thing, and we need to address this issue. So, with the effort of local university, like we have a university in Nepal with the veterinary schools, the student from there and some of the foreign student from US, we both of us collaborate and uh, able to we collaborate and we run a comprehensive elephant tuberculosis survey through which we identify the possible elephant which could be we identified the elephant and we started treating this elephant from 2008, and several elephant was benefited through this program. Like if you look on the elephant tuberculosis survey which had happened in 2006, and then it was the first comprehensive elephant TB survey carried out in Nepal. It was carried out all over in Asia, where we collected the trunk wash and collected the and run culture. Similarly, we have done the stack pack test, MAPIA test, and we also run the ELISA test for the tuberculosis. And also we look on the CBC, CBC and biochemistry, how the change is happening in blood count and biochemistry of the elephant. These are some of the known elephant dead case caused by the tuberculosis. We can see on the data there are the two cases in 2002, the Mohendra Gaj, and 2004, the Prem Kale. These are the first two cases which alarm us about the tuberculosis, and there was a wave of death till 2013. Then after, we do not have much mortality. We have most mortality was happening till 2013, and we have a breakage of 6 years. It might be due to the treatment we carried out, where we regularly monitor the elephant with the tuberculosis and do the treatment. So the mortality might have decreased over there, but recently, since 2019, there is again second wave of mortality is happening, where when some of the elephant died, we do the postmortem, we see the tuberculosis lesion, and from culture we have found that these animal are having tuberculosis. These are some of the examples of the TB cases which were seen in Nepal. These are the two elephant and the lungs of these elephant, where we can clearly see the TB lesions in them. These are some of the elephant. And recently we have published about the epidemiology and molecular characterization of the Mycobacterium tuberculosis, and including if they have any drug resistance association or not. These are the animal which was the elephant which was died between 2019 to 2022, and we have looked on the epidemiology and their molecular characterization of tuberculosis. Similarly, the one study we carried out in Nepal in 2018, 19, 20, where we look on the we look on the molecular characterization of the two cases of the tuberculosis dead during that period of the time, which show that the tuberculosis associated with our elephant, the one in the red and another red. These are the two cases of the tuberculosis which we see in Nepal. These are, one case was in lineage 1 and another case was in lineage 2. This these are the tuberculosis lineage we have seen in Nepal, we have found in Nepal, and it is more associated and which is more closely related with human. So we could tell that there is a chance that there might be a cross transmission. There could be a transmission of tuberculosis between human and elephant, and similarly between elephant and human. Similarly, in Nepal, we endorsed the first action plan. It was called as Nepal Elephant Tuberculosis Control and Management Action Plan, which was endorsed by the Ministry of Forest and Conservation of Nepal, and where the goal of this action plan was to prevent tuberculosis transmission to the wild by controlling TB at the captive wild interface, because the captive elephant is one of the entities which which is regularly engaged in the captive and wild interface, and they have tendency to transmit the disease from captive to the wildlife. So we addressed that issue so that tuberculosis could not transmit to the wild. Similarly, during 2006 to 2017, we tested around 254 elephant, and 61 elephant was treated during that period of time, where 17 governmentally owned, 41 privately owned, and 3 NTNC owned elephant was treated with tuberculosis treatment. And basically, there is still controversy whether there is like tuberculosis will it's good to treat the elephant or not, whether it what the drugs, there is a lots of side effect of the drugs, and even we are very much unaware what happened when we do the TB treatment in the elephant. But basically this photo will show that whether we'll know the elephant will the TB will completely heal or not. The thing is, it will extend the life of the elephant. This is the example of one elephant. Photograph on the top was from June 2011. It was before treatment, and if you it is the another one is around 2019. There is a considerable change in the like in the overall physical appearance of the elephant, and it extends the life of that elephant. Why TB is important? Like TB is important in Nepal because like if captive elephant are infected with the TB, they are generally used for the ecotourism. There is a chance that this elephant will transmit the infection to the rhinoceros, as this is shown in the photo. There is a chance it will transmit the disease to rhinoceros. Similarly, the breeding of our elephant is carried out by the wild bull. The wild bull come and breed with our female. So the chance that this bull can transmit the disease to the to the wild population. And similarly, as there is a close interaction between human and elephant, there is a chance that the disease can transmit to the local people. And similarly, we can see the elephant and local cattle are livestock are grazing together. The chance the transmission might happen between elephant and livestock as well. So these are some of the photographs how the elephant is closely associated with the human, and the transmission can happen to the public as well, and it's a very important public health issue. So we also reported the tuberculosis in wild rhino. It's Mycobacterium oryx associated tuberculosis, which is somewhat similar to the tuberculosis in the elephant. So there is a chance that tuberculosis might have been transmitted from like from elephant to rhino. It could be a possibility on that aspect as well. So another important disease we come across in our elephant is elephant endotheliotropic herpes virus. So everybody knows about the impact of this disease globally as well in our range country. In Nepal, till now, we have got 13 cases. Like if you look on the overall demography of the cases, like we have the green one is the one we able to save with our treatment protocol developed here. And most of the if you look on recently there is a huge wave of infection going on in Bhatia National Park, and many calf are dying over there. And like in Chiton National Park, in our breeding center, we have till now 80 calf has born over there, but we have got only 3 cases of herpes ESV virus in our breeding center. So it's very important to know about the cross immunity happening in the group. So it would be a good place to study about that aspect. And interestingly, since 2019, we do not have got any cases now. So we cannot tell that it has completely stumped out, but somehow we do not have got an infection. But most infection has happened between 2015 to 2019. In this 5 years period, we have got around 9 cases of EESB. So these are some of the cases we have seen in our elephant. So we have developed a treatment regime in Nepal, which we generally use the famciclovir like 12 mg per kg body weight. This is the first case where we use all of the case we use the per rectal, but we can like if the elephant feed orally, it's easier. But if they do not feed, we can give per rectally QID for 2 days. It's per and TID for next 2 days and BID for remaining 17 days. We do the treatment for complete 21 days. We can give rectally as well orally, but the treatment we followed here was one case, like most of the case we have done rectally, and one case we have done oral treatment. Similarly, we give NSAIDs, meloxicam, 0.6 mg per kg body weight, IV for 5 days. And first one, we have given the fluid therapy in one cases, but generally I do not prefer to give a fluid therapy because when we give too much of it's my way of treatment. It could be controversial from other, but I generally do not prefer the IV fluid therapy, but better to give rectal fluid therapy, give plenty of the water to the elephant calf and just motivate the elephant calf to drink lots of water. This way we can do, and also we give the antibiotic. One case is where we give too much of fluid, it developed the edema on the neck area, and the elephant calf was unable to respirate. So we have to give the diuretics, and it helped to normalize the calf. But with my experience, I can tell that it's better to give the plenty of the rectal fluid and put water inside just in front of the calf and motivate the calf to drink that water. So, and if the calf take a water, we generally do what we do is one of our mahout will be together with the calf when they are infected with ESB, and they just when the calf start to pull the water with the trunk, take with the support assist of mouth, put that trunk inside the mouth, elephant mouth, so that they can drink the water. Similarly, these are the first survived case from our treatment protocol. This is the first day. Second day, it has developed the clear ESB symptoms with the mandibular swelling and the cyanotic of the tongue. And in third day, it was its neck was swollen and it was unable to urinate. Then after when we give like diuretics, it urinated and it's get better. In fourth day, fifth day, it start to get back to the normal way. But we still continue the treatment for 21 days. Another important, most important, I think globally, it's the most important problem with the elephant, the captive elephant or working elephant is the food affection, or even the zoo elephant. Food affection is the big issue. Generally, some of the food affection we could see the overgrown pad, overgrown nail and pads, and osteomyelitis. Similarly, fungal infection. Generally, our climate is very much tropical, and we have got a lot of rainfall, and our our monsoon is very rainy. So we got so many we got so much rainfall that mm, our place will be very like swaggy or swampy, so that the elephant get fungal infection through that process. We have found many cases of this type of fungal infection in our elephant. So we have got a cracked nail, cracked front nail, split nail, and these are the different scenario we could see in the food affection we see in our elephant here. And what we do is we try to address that problem since the last 3 years. Now we generally do the regular nail trimming and food trimming work, but with the support of Cathy Adamson Conservation Fund and MacLear International Consulting, we get a international export service about the food affection. So every November we run like a 3 week health camp, annual health camp with our elephant, where we look on the most of the food affection, and some of our zoo vet from states, they come here and help us looking on the different health condition and uh, support us through this program. And the good thing about this project, this program is our owner, the elephant owner, they are sensitized about the importance of the food, important about the shade they have for the betterment and better health of their elephant, especially their food problems and this type of the things. So we do the nail trimming, and like we maintain all the cracks, and we try to put on a better hygiene of our elephant nail. And very important, this is the one of the landmark case of this elephant health camp. Like we were very much, this elephant has this food problem, this sort of food problem like granulation tissues and this problem since last 20 years. And with the support of MacLear International Consulting, Mike, we try to address this issue. And and we this is the support of the international collab. This is the result of the international collaboration we carried out, international support and international collaboration. We started with Mike and our regular interaction. We think about doing the CO2 therapy to this food. Then this is a change we have seen on this food in just 15 days. So we plan to do this continuous treatment for more than 2-3 months and look on how the change is happening. And the Mahut, so this will be the one of the landmark case, especially to address some of the very worst food problem which we were not able to treat since last 20 years. So it is the benefit of international collaboration and health camp we are carrying out. Similarly, the parasitic infestation is one of the major problem. Like we have endoparasite. Generally, Strongylus is the common endoparasite we have. Like generally, our elephant, when they have the parasitic infestation, we have seen a problem that they have tendency to eat more. When they have a stomach problem, they eat more. And many time we come across the case of impaction and colic through parasitic infestation. But generally in every like alternatively, we do the treatment of fenbendazole for nematodes, and for the lice we give ivermectin, and for fasciola we generally give the tricalbendazole treatment. In in annual health camp, we generally give we treat the elephant with anti-parasitic medication also. So one of the another important cases we came across in our elephant is stomach bot also. Generally, this stomach bot we generally find out in our elephant after monsoon, generally during November. and October, November, our elephant came across the stomach bot condition, and they just stop eating. So we have a great problem identifying this infection as well, but we try to solve it with the ivermectin, and ivermectin treatment, and it was very much successful. So colic is another condition. Generally, our elephant, when they get this stomach infection, parasitic infestation, and also like when they feed mud, they came across with impact, and they have a colic. The symptoms of colic is frequently lying down, getting up, and frequent urination, lifting up hind leg, and resting or leaning against the wall, and they generally bite the tip of the tongue and just produce the special vocalization when there is a pain, and we give hyacinth butyl bromide for that treatment, for the treatment. But recently we have seen very much like a very increase in the case of impaction. Generally, before, when we have the elephant get impaction, we do the treatment, they will solve within like 2-3 days. But now we have seen some of our elephant, when they get stomach impaction or intestinal impaction, they have the symptoms, and they do not get healed for more than some of like 10, it takes 10-20 days for complete healing of this infection or impaction. Another important case we have done, I want to share, is about the vestibulotomy we carried out in one of our elephant in, I think it was in March 2023. And this elephant has previously also, it has had a dystocia, and we have to pull the calf down. But again, we do not, since our breeding is carried out by the wild elephant, we do not want to breed the elephant, but we get a unwanted pregnancy due to wild elephant. So this was when the elephant came in the she was in the started parturition, but the calf stuck over here for more than more than 3-4 hours, and we look on the blood profile, and it has calcium deficiency, and we give intravenous calcium therapy, but it did not work. And even we tried to induce the parturition with oxytocin, but it could not happen, and we waited for more than 8 hours. Then after, we finally decide to do the vestibulotomy, and we clean the vestibular area and give an incision and try to pull this calf towards the vulva, but it was not possible. We could not do that. So we make a big incision over here and pull the calf out. By doing that, we were able to take the calf out, but we do not know the calf was live or not. Luckily, the calf was live. So after about after the calf was so exhausted that it takes more than 4 hours to completely the calf could stand up. But unfortunately, the calf was not accepted by our dam, and it was not accepted. So when the calf we try to hand feed it, but after a month, it got enteritis, and it died. So we could not save it. So this is the post-surgery healing process happening, wound management, which was carried out. We try to, this is the first day. Second day, we try to close the vestibular mucosa with suturing, but it breakage, and we do the second intentional wound healing. So this is on the day 6, and day 18, it looks very good. And this is on the day 90 post surgery. These are the different stage of the different stage of the post surgery wound management. Now the challenges we came across with the captive elephant is most management in the captive bulls. Like in through our breeding center now, we have got so many captive bulls which born up from our breeding center. Now the big problem is how we can manage this captive bull. It's a big issue. I don't know how these captive bulls are managed in another range country, but here we are having challenge and doing that stops. And similarly, now we have seen recently 4 cases of dystocia in dam with hypocalcemia. Like they have lower calcium level in late stage of pregnancy, and during parturition they are not able to expel the calf, and we check the calcium, and when we give a bottle of calcium, it just give a birth of a calf. So how we can manage the nutrition of the dam so that the hypocalcemia can be managed will be a very important topic to discuss globally and with other range country as well. So this much from my side. Dhanyavad. Thank you. Any questions? Please, any questions which I can address?
Okay. Close panel. Dr. Amir, can you close panel?
Yeah, I'm here, but my network could be a bit I might get disconnect, but I am here. Please.
Oh yeah, okay. Yeah. Thank you so much. Thank you very much, Dr. Amir, for the presentation. In spite of the difficulties, you could able to do it. Thank you so much. We have couple of questions for you. Somebody has asked you which TB species you have found. Any studies which shows TB transmitted from elephant to humans?
The tuberculosis we have found in our captive elephant was Mycobacterium tuberculosis. As we have done the sequencing and alignment of these cases, and we have find out that our tuberculosis was aligned with the lineage 1 and lineage 2, which lineage 1 and lineage 2 of the tuberculosis. And also we have found that this lineage also human also share lineage 1 and lineage 2. We also if lineage 1 and lineage 2 of tuberculosis, most probably with that result we might say that it might have been transmitted from human to elephant, and we don't know whether it could be from elephant to human also. I'm like our publication could not, our research could not tell whether it gone from which side, but still it could happen from elephant to human also. As the publication is there, everybody can look on that as well.
Yeah, thank you so much. Dr. Asad has a question for you. Nice information. What are the main signs, symptoms of EEHV in elephants?
Generally, if you look on the signs and symptoms, generally, if you look on a textbook, the sign and symptoms of the ESV is the mandibular swelling, cyanotic of the tongue. When we get up to that stage, it is very difficult to save the calf. Generally, we in our case in Nepal, our calf goes to the grazing with their mother. So we trained our mahout that always observe the movement of the calf, whether the calf when they are going to the forest and coming back to the forest, how they are moving, whether they are moving in front of the mother or not, because we have so many calf, all of them, if they run in front of the mother means they are healthy. And if they start to walk together with the mother, then we think that the calf might be sick or they start to become sick. Our mahout help us on identifying the ESV quickly. And also if the calf is just behind the dam when they are going to forest or coming back from the forest means this calf, we found that this calf are sick with any sort of problem. So after that, we collect the sample, blood sample from the calf and check we run a molecular examination. So then after we'll find out whether uh, it has like ESB or not. That with that way we do not go up to the confirmative science, but we look on the other factors. Mahout also help us doing that, and we'll move look on the hard palate. Generally, in initial stage, we found that there is some sort of ulceration in the hard palate, and in a initial stage of viremia, the calf generally is unable to raise its trunk. So these are when they are just nursing, they are not able to raise their trunk. These are some of the things we observe, which help us in early diagnosis and saving the calf from this infection.
Yeah, thank you. Next question is from Dr. Ilayaraja Selvaraj. Can you please explain the CO2 therapy for a foot lesion?
I think everybody have seen the photograph of how we treat the uh, like CO2, how we do the CO2 therapy. This is the new technique we try to start in some of our elephant with chronic foot affection. One of the case I have shown with the community is that one has the chronic granulation since last 20 years. It is not getting healed, but some of our international collaboration with Mike McClare is a renowned person on foot affection. We try to do the CO2 therapy. In that, it's very simple. It's not a big issue. Just you put on a big plastic, put the foot inside that plastic bag, and give a CO2 therapy just 1 or 2 minutes only. Then you let that be inside the CO2 inside the bag for 30 minutes. Then you take out the bag out. By doing that, we found out that it will make the anaerobic condition in the foot and it just stop the growth of the bacteria. And as I have shown in the photograph, let's see if I can share it in my computer. Like, and we found out that doing this way, we find out that the foot was like this, and putting on the bag and putting on the CO2 through the tank for few minutes only, it will give a very beneficial effect. And this is the effect after only 10 days. Now we are finding, I am because I am out in the field, so I cannot have a new photograph, but now it got better. And initially it has very foul smell from the foot. The foul smell now stopped. So it is getting better.
Okay, yeah, thank you. You have a question from, you have a question from Mohammed Firdaus. Thank you for the nice presentation. Why the calf was rejected after the vestibulotomy procedure?
I cannot tell that. We tried very much to take a calf near to the dam, but she regularly kick him out. And even at the night time when calf goes towards the mother, it kick. So we cannot tell how why it do not accept.
Okay, Dr. Ilayaraja has another question. If any anaerobic bacteria will grow, how
We
to
haven't
manage?
found we haven't found it because we are not maintaining an anaerobic condition for more than 30 minutes. So I don't think the anaerobic bacteria there will be plenty of the time for anaerobic bacteria to grow on that.
Okay, yeah.
Let's see. It's a test case. We can tell only after. I even with this only I cannot tell anaerobic bacteria will not grow. Let's see if it gets successful. It will be a landmark, especially for the elephant foot affection. I think we are going to do a treatment for 6 months. After 6 months, we can tell it will completely heal or not.
Okay. Next one is from Dr. Bahar Bhaviskar from Nagpur. Is there any test and treatment to identify and treat the pregnant animal infected with herpes so that the cough is delivered safely?
I do not know about this aspect. Most probably, we have found that even I have done a surveillance of ESV in like our breeding cow, and we have found that our like female or our elephant are carrying the infection, carrying the virus, ESB virus, asymptomatically. And especially with our research also, we have found that the shedding of the ESB by female is more in second half of the pregnancy. First half, we do not find much shedding, but they are shedding even the pregnant female in second half is shedding the virus through its trunk. So I don't think it's a big issue. Even they are carrying the herpes infection, and it will safely deliver the calf.
Okay. A question from Dr. Bauer. Where do we send the samples for EHV surveillance? What samples?
We have our own molecular lab in our setup. We collect the sample and run it immediately. Like if we are going to look on a young calf, we collect blood sample. For the ESV surveillance in the adult, we collect the trunk wash sample and look ESV on that sample.
Thank you so much, Dr. Amir. Thanks for your replying to all the questions. That helps. Yeah, I think I think there's one more question here. Maybe if you can just stand from Dr. Palnivel Rajan. Sir, CO2 incubator is essential for anaerobic bacteria culture. In this case, how is controlled growth of anaerobic bacteria?
I already tell that about that aspect. We have just started the work. We anaerobic bacteria growth or suppression of aerobic bacteria growth. We only more focus on the food. If it get better, we'll be okay with that. But we do not care much about aerobic and anaerobic bacteria.
Okay, Dr. Christopher Sami has a comment on that. Daily soaking the food for 5 to 15 minutes with antiseptic solution, example, tincture of iodine of 2% formulation will eradicate anaerobic and anaerobic bacteria and lead to healing. In addition, trimming of uncontrolled overgrowing granulation should be trimmed. Okay. Yeah, that's a comment from Dr.
We have tried that several other treatment. All of that has failed. So we gone for this.
Okay.
For this elephant.
Okay, thank you, Dr. Amir. A wonderful presentation. Now I hand over the session to Dr. Heidi. So we are just in closing. Dr. Heidi, please.
Okay. Oops. Thank you. Really appreciate all the presentations. Thank you very much, Hamid. That was very interesting. Great presentation. And thank everyone for logging in and listening to the presentations. If you have more questions, please feel free to send them to us and we will share them with the presenters. And we'd be glad to continue on. We will have another webinar in February, I believe it is. It's every other month. That will be the next webinar. And so we will all keep you posted on this. And again, thank you to the presenters and also thank you to everyone who logged in to listen to the presentations. We really appreciate it. Thank you.